Hi guys, my name is John Harvey. I'm an undergraduate student at UC Merced, and right now I'm using NanoHub. So NanoHub is a very useful tool for my research, and basically what I try to do is I try to see which array or which nanostructure geometry works best at specific applications. For example, in solar cells, right? The whole point of solar cells is absorption. So you want to absorb as much light so we can transfer it into energy. Now, in DDS-CAT, which is one of the tools in NanoHub, we could see which array or which structure or which configuration can give the most absorption, right? And so that's one application. Another application is visualization, scattering. Scattering is very important in molecule visualizations, whether it's in SEM, TEM, or even like biomedical visualization. So we want to increase scattering in that. And so if we want to increase scattering, we want to play around with the configuration and see which scattering gives the best, or which configuration gives the best scattering. So that's what we want to do, and that's what we hope for. It's still fairly early in my research, so give me a year. So give me a year. I'll get there.
Right. Hi, my name is Leanne. My title of my project is Mechanical Forces Can Direct Vascular Cell Fate. I am using a stem cell line that was derived in our lab that glows green and glows red depending on the types of genes that they are expressing. So we were able to tell if they're endothelial cells or smooth muscle cells live in the culture. I use a chip that's provided by our collaborator at UC San Diego, and these chips, they vary in stiffness, so they alter from soft to stiff, soft to stiff. And we were interested to see how these cells would differentiate in this environment. However, we thought that maybe the material was a little too toxic, so we used a control, which is a mature cell type, and we saw that these cells do indeed migrate to a specific stripe. And then lastly, I'm also interested in controlling cell-cell contacts. So I have a vascular leaf stamp that was also provided by a collaborator, and was able to pattern these cells into a branching blood vessel-like shape. Thank you.
My name is Kevin. My project is about cell migration in three-dimensional in vitro, driven by stiffness gradients. This is work done by, most of the work was done by Daniel Joaquin, my previous students. And so the objective of this project is how we try to track how cell moves alongside the stiffness gradients. So basically what we did was first made a PDMS wavy pattern, and then cover this PDMS with a matrix gel, and then CD cells on top of the gels. And we did a finite element simulation to make stiffness results. You can see here, and analyze the stiffness gradients using this simulation, and CD the cells on top of the gel. Image of this can be seen here with SEM image. And the results you can see when cells were distributed randomly, they moved toward the highest thickness point of this matrix gel, so that after 24 hours, they make a cluster alongside the stiffness, highest stiffness points. So we analyze their movements, and this graph shows the X point versus mean velocity of cell. And you can see that when we analyze with the stiffness gradient versus cell movement, cell velocity, and you can see they tend to move faster when they feel the stiffness, the highest stiffness gradient. And we did similar experiment with other cell types, and even though the morphology is a little bit different, they also have the same tendency. So our conclusion is cells can actually feel the stiffness gradient, and their velocity is related to this stiffness gradient. Okay.
Hi, I'm Z Chen from Washington University in St. Louis. So we are interested in studying mechanics without muscles, so the fast motion of the Venus flytrap and bio-inspired robotics. So the Venus flytrap is a plant. It doesn't have any muscles or nerves, but it can move remarkably fast. In a fraction of a second, it can close to capture insects for nutrients. So if you take a look at the flytrap closely, there are several sensitive hairs on each of the lobe. And so if you touch it once, it doesn't move. But a second touch within a window of 25 to 30 seconds, it will quickly snap through to capture whatever triggers it. And so this is a pretty smart mechanism that makes sure the plant doesn't waste energy on capturing unworthy things that are randomly fallen leaf. There have been a number of researchers, tackling on this problem from different angles, and the studies on flytrapping also inspire, biomimetic designs of engineering structures for applications. So, in terms of the change of geometry, we noticed that in an open state, the Venus flytrap actually has a positive Gauss curvature, meaning the product of the two principal curvatures is positive. And in a closed state, it's also positive, although each of the principal curvatures flip sign. So the product remains positive. And this is actually similar to this toy popper. So you can see that, in a stable state, it's doubly curved, and the Gauss curvature is positive, and inverted, and it becomes another shape with positive Gauss curvature. You wait for a few seconds, and then it would jump. So it's like a toy popper. So we think the change of geometry is pretty similar here. So we want to understand, are there any similarity between the mechanics of these different structures? So there's some scientists from Forte and Mahadevan, they proposed a theoretical model which takes into account the bending energy and stretching energy in this plate theory to interpret the Venus flytrap's bistable behavior. And what's interesting in the flytrap, though, is if you cut it along this vertical direction, then it's not surprising it will curve inwards. But if you cut it along the horizontal direction, surprisingly it will curve outwards. So that seems to suggest that the Gauss curvature locally would be negative. So it would have formed a saddle shape instead of doubly curved hemispherical shape. So to interpret this dilemma, we add another term which basically characterizes how this edge area of the flytrap deforms when you go from an open state to a closed state. So basically you can imagine that the edge area will undergo a dramatic deformation. So that creates the energy barrier. It's pretty much like in this 1D analogy, going from this to that shape, you have to push against the springs attached to a wall. And so by adding this term, we found that the ratio between the end configuration of the Gauss curvature and versus the starting configuration is about 1. It's very close to the experimental measurements. Furthermore, we also examined what has not being paid much attention to is what connecting those two leaves. So most researchers focus on the dramatic deformation of leaf, but we think the midrib connecting two leaves are also important. So we found that we can trigger actually just one of the leaves to close, so it becomes an asymmetric shape without touching the sensitive hairs. And now we can trigger the other lobe to close. Now it's symmetric, but it's not completely closed until you change further trigger it to close the angle between them. So we found this unconventional deformation mode in the flytrap. So basically, there's a bistable behavior on the leaves and the change of the angle between them, and it's a combination of them that will lead to full closure. And based on the principles learned from the flytrap, we also designed biomimetic flytrap robots here. So the idea here is one mechanical touch, it doesn't close the flytrap robot, and the second, it will close. So one touch, it only deforms locally. So this basically mimics the fly trap behavior, but through a purely mechanical mechanism. So we hope our study can also inspire the design of artificial muscles, because the fly trap is, basically in the plant kingdom is probably the closest one to the muscles. We hope that the study of mechanical principles in fly trap can also help engineers come up with novel structures for engineering applications. And also interesting question can be raised from the evolutionary biology point of view, like how did they evolve these very smart mechanisms in the first place? How did they know the existence of these live insects, live food? And there are also other closely related species that exhibit similar but different trap mechanisms. So how do these trapping mechanisms evolve in plants, and how are the mechanisms conserved? So, yeah. And our research has been supported by Society in Science of the Branko Weiss Fellowship and the American Academy of Mechanics Foundation Founders Award.
I am Ram Godawali, and I work for Palmer College of Chiropractic, and we have a research center dedicated for research. And there are several forms of chiropractic procedures for treating neck pain. One of the treatment procedure is the patient is in a prone position, and the doctor of chiropractic holds the contact at C4, C5 or C6 vertebra and applies a traction force. And this procedure is called manual cervical distraction, or also known as flexion distraction technique. So this is a funded study, and we are from NIH, and we are collaborating with Loyola University, Swiss School of Medicine, and also Heinz VA Hospital. So the objective is to measure the changes in the intradiscal pressures in the lower cervical spine during manual cervical distraction procedure. We took 9 unembalmed cadavers with a head, neck, and trunk, and inserted pressure sensors into the C45, C56, C67, C71 discs. And when 4 chiropractors, experienced chiropractors, were applying this procedure, traction procedure, we recorded the changes in the intradiscal pressures, and these are shown here. As the doctor is applying traction and releasing, traction and releasing, traction and releasing, the pressures decreased as a function of the treatment in all the 4 discs. And as the force is increased, the pressure has decreased. So this is the mechanism by which they think this is helping the patients relieve the back pain. What is happening is when you apply a traction, there is a pressure gradients happening, and the nutrients flow back and forth. That creates an opportunity for the disc to regenerate. And another thing is when you apply a traction, any disc bulge has a tendency to reduce the disc bulges. So when we measure the forces, they can be as high as 120 newtons, and the mean intradiscal pressures were as high as 168 kPa decreases. And the doctors repeated this procedure 3 times. All these 4 doctors, when they repeated 3 times, how consistently these are the intra-class correlation coefficients. They are pretty good, from anywhere from, except one case, 0.83 to 0.99. Only one case, 0.64. But traction forces, 2 of the doctors are applying consistently 0.93, and 2 of the doctors only moderate consistency. So we validated the decrease in the intradiscal pressure and the consistency in which the doctor is delivering the treatments. And we are also doing a clinical study. We are finishing up a clinical study, writing manuscripts. The goal is to find the optimum traction forces. Our clinical study did 3 treatment groups: low force, medium force, high force. Is there a certain threshold of force that gives optimal? So is there a certain 20 newtons or 40 newtons, 60, 80? Which of these forces will give a better treatment outcome corresponding to the decrease in the pressure? That is the future research. Optimizing the treatment is our goal. And the purpose of my coming to this seminar is there, when you apply these type of forces, how the disc cells are reacting or responding. Is there a potential for the disc to regenerate? Thank you.
Hi, my name is Yi, and I've just finished my first year in the PhD program in Professor Ryan Bailey's lab. I'm from Department of Chemistry. So here my poster is about a droplet-based microfluidic device for nanoliter scale chromatin immunocapture, or we can call it nanochick for short. Basically, it is for epigenetic study. Epigenetics, it's the reason, like, even if your siblings share the exact same genome as you guys will still have different phenotypes. It includes the DNA modifications and histone codes. So it will control your gene transcription and DNA synthesis and even repair. It has something to do with both beneficial and bad cellular progresses, including aging, carcinogenesis, and cell-fate function. So this is really important. If you can find some information behind the epigenetics, it will definitely be helpful with either clinical applications or just like knowledge. So traditionally, this part will be done by a technique called chromatin immunoprecipitation, or CHIP. So we will first cross-link your DNA to the histone protein wrapping around your DNA to make sure that both of them are still a group, even if I do a lot of dealing things to the cell. And then we'll lyse your cell, sonicate it, and now you will get your fragment chromatin. Now you're adding your specific antibody-modified magnetic beads to pull down your interested histone, of course, as well as the DNA. And then we will protease the proteins, left only DNAs around, so that then we can run the PCR to get the sequence of your DNAs. So this is the whole technique we're going to use. The problem with this traditional and widely used technique is that it requires large cell populations as input, which is not good, because nowadays we do a lot of researches on cancer cells and those rare cells. Normally they won't be so much. So let's say you struggle a lot to get your cell samples. You want to run a sequencing to find out something behind that. But then you realize that this technique cannot do your job with so small cell samples. This will be really disappointing. So we want to apply all of these advantages of droplet-based microfluidics to move every single step onto a chip, so that it will be automatic and it will save your sample size. And again, it will give you even better results. So basically now I'm doing AIM-1 and AIM-2 independently to do proof of concept drops. It's an ongoing research project, so the results are not so good, but still we make progresses, and means that this idea works. Here's my enzymatic monodinucleosome preparation steps. Basically, I do online cell lysis and cell digestion, and then I quench it with EDTA. Then I run gels to see whether I really get these nucleosome digested into pieces as what I want. And this is the quantification results. They are not in the correct orders as what I want, because you can see with higher cell samples they get like even like like even a lighter bands, but with medium sample size, we have the best results. But still, this shows something good, which is for all of these on-chip dealing methods, we have more mononucleism. This is what we want. We don't want di and trinucleism because that means all of these nucleism I'm interested in are still get together. We don't digest them completely. So I will do optimizations in the future, try to get at least the same similar results as off-chip. And here is for immunoprecipitation, which is one of the key features of my AIM2. So I want to concentrate my magnetic beads so that I can do the following PCR with my targeted DNA. And here you can see with a droplet with magnetic beads coming over, the beads will be pulled down to the bottom of the droplet by the magnetic, and then all of the beads will be at this part. So when this droplet hit on the side wall of the channel, most part of the liquid will get split onto the upper channel, while what I'm going to get will be the concentrated droplet with all of my magnetic beads, and this will be collected at its outlets for future detection. So basically now, I've already proved that my on-chip Crabtree shearing directly from cell with smaller populations is successful now. And my concentration magnetic beads has also been successful. So what I will do is to, of course, optimize all of these steps, and I will integrate these two parts so that I can finish my AIM1, and then integrate this AIM1 with my AIM2 steps, so that finally I will get my ultimate objective. So that's all.
Hi, I'm Rachel Hitano, and I just finished my first year in a PhD program at UC Merced in Dr. McCloskey's office. And my project is on generating a human embryonic stem cell derived cardiomyocyte patch in order to help repair a cardiac infarct. So your heart, when an infarct occurs, the cardiomyocytes don't regenerate. So a problem is that you now have the scar tissue, which impairs the contractility functions of the heart. So what we want to do is we want to derive cardiomyocytes, and we want to pattern them before we put this patch into the heart. So what we did is we took a protocol that we've already published, which takes the human embryonic stem cells and drives cardiomyocytes, and then we worked on adding different mechanical factors to this system. And in this case, we added nanotopography in order to align the cells. And in the future, we're going to work on adding the mechanical stretching force in order to help them become a more mature phenotype. So in order to take what is a aligned cardiac patch and layer them together, we're using p-NiPAM. And what this is it's a thermoresponsive polymer, which when it moves from 37 degrees Celsius to 23 degrees Celsius, the layer of cells that's on top of it lifts all at once. And in this way, we're able to layer the aligned cell sheets, and we've done this with endothelial cells and with smooth muscle cells, and so next we'll be working on doing it with our derived cardiomyocyte sheets. So far I've worked on deriving the cardiomyocytes and characterizing them and making these aligned monolayers with the sheets, and in the future we're going to use this method for the mouse infarct model in order to help with repair after infarct.
Hi, my name is Kiran Chauhan, and I am from UC Merced, and the title of my poster is Phylogeography of Euphoria victoria using mitochondrial cytochrome C oxidase subunit 1, CO1. So the main idea of my research is collecting jellyfish from different locations, from different time periods, and assessing whether there are genetic variations or similarities, whether there's genetic structure between these individuals, between the different jellyfish. So what we do is we collect these jellyfish. I don't do that, my grad student mentor does that, but they collect jellyfish samples ranging from Newport, Oregon to Pillar Point, California. And once we get back our samples, we pretty much we do DNA extraction, we amplify CO1, which is a common genetic marker used in genetic studies. And once we amplify that, we send out our sequences to Berkeley. Once the sequences come back, we pretty much go through a whole long process of cleaning sequences, making ARP files. Once those files are made, we separate them out for different types of comparisons. So one of the comparisons we make is temporal, latitudinal, and longitudinal. So these different comparisons are made, and the statistics that we generate are FST values, p-values, and FST values. And the values that are bolded, they show that there is some significant genetic variation occurring between certain types of individuals from different locations. And when these results are come together, we get FST values. And the most significant value that we found was temporal genetic structure. So individuals that were collected in July differed from individuals that were collected in September. And that was pretty interesting, but we need to do more studies using a more specific marker to make sure that our studies are significant, and we need to find more data points. But that's pretty much our research, and I think it lays the groundwork for further studies. I find it really interesting that I find jellyfish really interesting, and the fact that conditions that are harmful for us, like pollution, are actually optimal growing conditions for jellies, and they thrive in polluted waters and in acidified waters. So maybe studying their genetics can help us control them in the future. That's not our study, but that's something to consider for future. Thank you.
Hi, this is Jennifer Rico from the City College of New York, and I've been working on medulloblastoma chemometastasis using different growth factor concentrations. So what we did at City College is that we studied medulloblastoma in a wide range. We look at different growth factors that are well known in brain cancer, but also a growth factor that was found to be a very chemoattractant for medulloblastoma. And what we did is that we compared the chemotactic potential of FDF1 with EGF, HEF, and PDGF beta-beta. And the first thing that we do is that we look at different nano concentrations of the growth factors, and we look at them through a transwell assay, and we found that EGF, HEF has a concentration dependency as opposed to PDGF and FDF1. Then we look at how the receptor expression plays a role in migration. So from the results that we obtained from the transwell assay, we look at the highest concentration at which these cells migrate, and we stimulated with their respective growth factor receptors, and we found that EGF have the highest receptor activation. It was a surprise for us that CXCR4, the growth factor receptor of FDF1, also showed highest receptor expression. So what we did is that in order to understand what's really going on between these two ICC and transwell assays, we use a microfluidic device that we designed and developed in our lab. We look at how different concentration gradients are affecting the migration of the cells. We look at how these cells, what type of migration these cells are experimenting when we are stimulating them with EGF and FDF1. And finally, we look at what is the maximum accumulated distance of these cells that travel along the channel. And from these results, we found that cells stimulated with EGF, they have a chemotactic migration, they have a directional migration towards EGF, whereas in FDF1, these cells, they're moving back and forth just because they are feeling the gradient, not because they are really attracted to the gradient. So from this work, we have concluded that EGF induces the largest number of cells to migrate, has the highest receptor activation, and makes cells to move at higher gradients in a directional fashion. And we believe that EGF should be looked as a potential chemoprotective factor to treat medulloblastoma metastases.